Well, good morning, everyone, and welcome back. I haven't seen you since last year, which was only a couple of weeks ago, but it feels longer. I hope you had a good Christmas break, and you were obviously safe enough that you're back. And amid this ongoing pandemic, I think if we have a distance here, we have plenty of room to spread out so we can take our masks off. All right, so we're getting into the second semester of the Masters in Bioethics here at St. Thomas University. And the two courses that we have for this semester are the Beginning of Life Bioethics and End of Life Bioethics on the human side, all right? So as always, begin with a little prayer. In the name of the Father, the Son, and the Holy Spirit. Come, Holy Spirit, fill the hearts of your faithful, enkindle in us the fire of your divine love. Through Christ our Lord. Amen. Okay, let me see if I minimize the lights. You still have enough light there for your notes? Yes. And the screen, you can see it better, right? Okay, so this semester, we're going to be focusing big time on the human life issues at the beginning and at the end of life, the human life bioethics, okay, which is one of the two large pillars, if you will, of bioethics today, is the human life issues. And the other one is environmental bioethics, which we'll cover in the summer, coming up right after the semester. So we're getting deeper and deeper into the bioethical issues, first on the human life side and then on the environmental side. The third aspect of human life bioethics is going to be healthcare bioethics, which is kind of in between the beginning and the end of life, all right? And healthcare bioethics will be in the last course, the last semester, which will be the fall, will be your second fall. And then you're done after that. So you'll be graduating, God willing, by December of this year. We just had the previous cohort that graduated a couple of weeks ago, actually. So it moves fairly quickly. All right. Again, I always like to ask if there are any questions or comments beforehand or at any point. Just stop me and we'll address it. Any lingering issues? No, I think everyone is enrolled on the technical aspect, on the administrative side of things. Everyone is enrolled in the course, and you can get into Canvas and see, I see on the roster all your names. So that's a good sign that you're all enrolled in the course.
I have a question.
Yes, Chris.
No, George.
Jorge, sorry.
George. So is these two courses that we're taking this semester, they're gonna be split in half, as in from now to like February, yes, we do beginning and then we do end of life?
Yes, thank you. Thanks for the clarification. Yes, because on the schedule, these two courses may appear as full semester courses, you know, the 16 weeks. And then one is scheduled, say, for Saturday morning, and the other one is scheduled for Saturday afternoon. However, because we go for 3 to 3.5 hours typically on the lectures, then what I do is, in practice, the first half of the semester, the first 8 weeks, will be on beginning of life. And the second 8 weeks will be on the end of life. That way, we don't have to come back in the afternoon. We get it all done on Saturday morning, and we keep that same schedule routine as we did last semester, okay? So yes, thanks for the clarification. That's how we work it out, even though for the purpose of fitting it into the official schedule of the university, I cannot schedule two courses at the same time. So I have to give it a different time frame, and that's how it's worked out, okay? But yes, bottom line, we actually have 7 modules for beginning of life and 7 modules for the end of life. And that means that also in the spring, we lose at least one Saturday, which is going to be Holy Saturday during Holy Week, right? Good Friday and Holy Saturday. Obviously, we're not going to meet on Holy Saturday. It's a very sacred moment of reflection and so forth. And so we're out that one week, and that's why it's 7 modules instead of 8. And then also I'll leave one Saturday empty between the 2 courses. By the time we finish this, first course, there'll be one Saturday free, which of course I'll be at the forest doing the management thing. So if you want to use that Saturday for your internship hours, again, keep in mind, I'm giving 3 for 1. In other words, 1 hour at the forest will give you 3 hours of internship. Then you can use that particular Saturday where we'll have no meeting. All right, and that would be in between the 2 courses, so halfway, about 7 weeks from now. While I'm on the internship, just kind of a stream of consciousness here, I want to mention that I encourage you to take advantage of the opportunity that I'm offering of this 3 for 1, in the sense that, you know, we're in a second wave of the coronavirus. And so the rate of infection is going up again because we know it's a winter virus, it's a flu-like virus, so it likes cold and dry. And therefore, it's still difficult, if not impossible, to get internships elsewhere. People, companies, and institutions are not accepting volunteers right now. It's just too risky. So normally, these institutions are the ones who would be available for doing the internship in this program, which are typically hospitals or clinics or the Respect Life office, or even nature rescue places like there's one called Pelican Harbor here in Lake County and so forth, but those are difficult to come by right now. Even in the parishes, some people have done internships in catechesis or workshops on bioethics at their own parish, for example, or with other groups like a youth group, etc. But most of those things are also shut down for now. So here's an alternative, which is to do some restoration work at the forest. And again, in the sense that the advantage is that you get 3 internship hours for 1 hour out there. And I can tell you, one of you has already done so many hours that he's practically done with his internship, okay? And it doesn't have to be Saturdays either, because most Saturdays we're gonna be taken up with the program now for the summer, with the 2 courses. But during the week, I'm also there, and I will have some undergrad students with me in small groups. So if you have any time during the week, we can work it out, all right? If you have some free time during the week and you wanna do that. The next advantage, and I think I'll stop about the internship with this, is that because we are in the winter time, it's not hot and humid outside, right? And so the forest is a little more pleasant to work because it is physical work, and therefore is not hot and humid and sweaty like it is typically in the summer. So that's another advantage. Essentially, it's like an air conditioning out there. It's like an outdoor air conditioning. Okay. Anyone have any question or comment about the internship overall or in particular? It's still one of the courses, and we cannot get rid of it because, see, 30 credits is the minimal for a graduate program or a master's program. It's 30 credits. And the internship is worth 4 credits. So if we get rid of it, we're going to be below the minimum requirement for the master's degree. All right, so we have to keep the internship, and you have to get creative about it. So this is one way of doing that.
Professor, right. I have a question.
Yes, go ahead.
Is there a rubric that is associated with the hours that we need? Essentially saying what we can and can't do for the hours?
Right, yes. It's in a syllabus, but it's pretty wide open. Basically, it means that the student, as long as the student engages in some aspect of dialogue in bioethical issues, either human life issues or environmental issues. And this could be done either formally or informally. For example, formally would be to give a workshop, right, on bioethics. Informally is just a discussion group, maybe with family and friends. But the important thing about the internship is to log the time, right?
Okay.
And I believe I have sent you, did I send you an Excel spreadsheet?
Yes.
That was basically blank with 2 columns. The first column is the date of the engagement of the dialogue, and the second column is just the hours. And the hours, take it down to 0.5 to half an hour, and to the nearest half hour. So just adjust the, like for example, if you give a talk for an hour and 20 minutes, well, round it up to an hour and a half, which is 1.5, right? And so when you turn it into a decimal point, it's easier to add in Excel that whole column. So does that answer it, Chris?
Yes, it does. Thank you.
All right, you're welcome. And of course, here at the graduate level, we're on the honor system. So I trust that if you put that you had a conversation on bioethics for 2 hours, then I trust that that actually happened because we're on the honor system, okay? So yeah, it's part of the professional integrity that we hold.
Gotcha. All right, thank you.
You're welcome. All right, anything else? Let's go forward then. All right, so in the beginning of life issues, remember again that the way that I do bioethics is like a 2-step thing, really taking off on the word itself, on the noun, bioethics. It has a biological component or a natural component, and then it has an ethical component or philosophical reasoning, right? And so we look at nature first. We look at what is happening, the natural facts, what is happening in nature, and then we analyze that and we come to some ethical conclusions based on what nature is doing. And that allows us to do what we call an objective bioethics. Objective meaning that is based really on nature, what we call natural law. Now, we can add a 3rd layer. So the first layer, let's say it's nature. The second layer is philosophy. And the 3rd layer, the deepest layer really is theology. Even philosophy is, we say, is the tool for doing theology. It's just that theology takes it to a deeper level, to the full level of the big question, which is the question of meaning. Where do we come from? Where are we going ultimately? And what is our purpose here? What are we doing here? And the fact that, again, a biological fact, that we have the capacity to ponder, to raise these questions. No other creature has that capacity that we know of, okay? Yesterday I was actually doing some work at the forest, and I looked about there in the field, and there was an iguana going by. And that iguana has no concept of who it is or where it is or anything like that. It doesn't really ponder its existence. The iguana was just by instinct looking for food, right? And that's the way it is. So because we have this capacity, it behooves us to exercise it. And that's what we're doing. So we can come to some reasonable conclusions when we look at nature, because ultimately, this is at the level of belief, of course, and the theological level, that natural law is a reflection of divine law. In other words, that God himself has an order, an internal order, and that order is manifested, is projected in the creation of the universe, including this little tiny bead that we call planet Earth, okay, within the solar system of the Milky Way galaxy, etc. And as far as we know so far, the only planet where life exists, at least organic life in the way we know it, as we recognize it, right? Okay, so basically, the first half of this course then is going to be setting the groundwork, the biological groundwork for answering a key question that becomes a bioethical question. When does life begin? In general, for all species, and in particular for the human species, right? When does the individual life begin? And that is going to allow me to do a brief review of last semester on the course on fundamental principles in bioethics. So really I should reverse these slides like that. Yes. Okay, so remember that this program is a philosophical and theological analysis of humanity, right? What we call a theological anthropology. So this phrase, Judeo-Christian theological anthropology, is really loaded. Those 4 words or 3 hyphenated words, it's really loaded. And we can start backwards and move forward, move from right to left. So anthropology is a study of the human, right? Anthropos is a reference to the human, and logos, logos from the Greek word or knowledge or science. So it's a study of us humans, and it's theological because we're putting it within the context of a belief that we are created in God's image. We're created in God's image, and that makes it Judeo-Christian because that belief comes precisely from the Judeo-Christian tradition. Okay. And you notice that in last semester, I started by looking at the possibility of evolution and then the actual fact of evolution with the evidence that we have there. And we can distill it to 2 major aspects, especially when we look at human evolution, because biologically we're mammals and we're part of the animal kingdom and so forth. So organically, we are not exempt from the forces of evolution, right? Remember, migration, drift, selection, and mutation. So we're not exempt from those forces. And we can distill the evidence for evolution on the human, in particular, is with 2 facts. The fossil record, we cannot deny the mineralized fossils, skeletons, skulls, and fragments that have been found and have been dated and have been located in particular regions of the world. And the other aspect is at the molecular level with the phylogenetics of our DNA, connecting us with the tree of life, with common ancestry, okay? And so we looked at the origin of our species as a whole, not individually, but as a whole. And that's why it's the phylogenetic origin, all right? Phylum is a reference to the group. In fact, one of the categories in Linnean classification, in the scientific classification, you know, is the phylum, right? Right after kingdom, phylum, then class, order, family, genus, species, remember that? So phylogenetically means where did we come from? How did we start as humans, as Homo sapiens, as a group, And that we can also break down 2 levels, which is at the level of species and at the level of genus. So at the level of genus, Homo, we share that with other hominins, remember? At the level of genus with at least half, maybe half a dozen to maybe a full dozen other Homo genus, all of which are now extinct. Remember, Homo erectus, Homo habilis, Homo neanderthalis, and so forth, right? These are fossil records, we cannot deny them. They're now extinct, but they have been dated and located to a particular time and place on Earth. And we can say that at the level of the genus, Homo, the time span is anywhere between 1 to 2 million years ago, right? And at the level of the species, sapiens, 1 to 200,000 years ago. Okay, remember mitochondrial Eve and chromosomal Adam, all that's coming back, okay? All right, so that's phylogenetically. And again, in the big picture, the main purpose of that course last semester here on the ground was to be able to reconcile the biological fact of evolution, including for humans, and the theological belief that we are created in God's image and that we can actually dovetail those two without having to deny or reject or ditch either one. So that if we are scientists and we allow the data to speak to us, we can also be believers. We don't have to be agnostic or atheist, right? And if we are believers, especially in the Judeo-Christian tradition, we don't have to reject science or evolution because it matches up. In fact, I did a nice math job, I believe, with Genesis 1 and 2 on the sequence of the order, beginning with the Big Bang. In other words, the creation and evolution of the universe, and then of life within the planet, and then specifically with the human. All right. So that was, in a nutshell, what took us 16 weeks to do that semester. All right, now we're going to shift gears a little bit, and we're still going to be looking at origins, but we're going to look at the individual origin of the human, each one of us. And that's what we call ontogenetically, all right? So remember, one of the added values of this program is that you'll pick up a little Latin and Greek on the way, because again, our Romance languages and modern languages, many modern languages, especially in the Western Hemisphere, are based on Latin and or Greek. Certainly the Romance languages, which are Spanish, Italian, French, Portuguese, and Romanian, but also the Teutonic languages like German and English. So ontos is being, individual being. And so when we talk about ontogenetically, it means the origin of each one of us at the individual level. Okay, so that's the main purpose of this course, is again twofold. First, to establish biologically when human life begins, and then to do the analysis based on that, what are the bioethical consequences of accepting the fact that human life begins essentially at fertilization, at completed fertilization. And so to understand completed fertilization organically, we have to do a little bit of biology and genetics. Okay? And so this program is kind of broad range, and every cohort, you know that you're the 5th cohort already. The program has been going on for 5 years now, and every cohort is a little different. I've had cohorts where there have been medical doctors, nurses, teachers, priests, deacons, wives of deacons, CPA, housewives, who else has been on the course? Students applying to other, graduate students applying to other programs, just all over the place, okay? And so what happens is that I have to cover the background on both sides, on the scientific side and on the philosophical / theological side, all right? So this particular cohort happens to have, most of you have a scientific background. So part of this lecture is going to be, these 2 courses, part of it is going to be a little boring to you at the beginning, because this is stuff that you have seen or you should have seen at your bachelor's level in science, right? But some of you don't have that biological background, so we just have to be patient with those people, and so that we can bring everybody on board. On the other side, when we get to the more philosophical, theological side, some students are more competent in that because their background is in that, and less on the science side. All right, so that's where it gets more challenging or exciting, hopefully for you, because it will be more new material. You follow? Hmm? Okay, so having said all those preambles, let's move forward now. Today's lecture in particular, we're going to look at reproduction and some DNA genetics to understand origins, all right, at the individual level. So going forward, we see when we observe nature, we notice that there are essentially two modes of reproduction that we have discovered on this earth, on this planet so far. And so far of the, how many species have been classified so far? About 2 million, right? About 2 million. It's estimated that they represent 5 to 10% of all the species that actually live on Earth. These are some numbers that I threw out last semester, but it was, I think, towards the beginning, so maybe you don't remember too well. Today, approximately 2 million species have been classified in the Linnean classification scientifically, right, with genus and species, which means that they are placed in a particular place in the phylogenetic tree. And 5 to 10% means that there's an estimated 10 to 20 million species out there, which means that we have not discovered most of the species that is estimated to be living on Earth today. Now, why have we not discovered those species? Okay, every now and then you see in the news, even in secular news, a report of a monkey that was discovered deep in the Amazon jungles, or some other creature, some other lizard that was discovered in the Congo Basin or somewhere hiding under some log in a forest, okay? But that's rare. In other words, that's at the macroscopic level that we can actually see this new species. That's fairly rare. Most of the species that we haven't discovered are estimated to actually be microscopic, all right? So they can be right in front of us and we don't see them. But further, not only microscopic, but typically at the ocean floor, 1 to 2 miles below the surface of the ocean, whether the Atlantic or the Pacific. And it's one of the frontiers where we have explored the least so far. So the Earth's surface, we have explored quite a bit of it, but the ocean floor, deep in the abyssal zone that is called, because of the pressure, the tremendous pressure that is down there, just crushing pressure, we have gotten some instruments down there, but no humans yet, okay? So we're dependent on the instruments. It's a little bit like exploring the surface of another planet, including, or even something like the moon. And in fact, it's actually a little more difficult than exploring the surface of another planet. The challenge of another planet is getting there. But once we're there, typically we don't encounter the pressure that we encounter at the bottom of the ocean. Okay, so those instruments that we send down there and the equipment, including the remote sensoring that needs to happen for the instrument to work, it's quite sophisticated. So that's the main reason why we haven't discovered all these other species, but typically they're considered to be microscopic at the bottom of the ocean. And we'll discover them someday, I suppose. Back to the surface of the Earth where we are, we notice that these are the two modes of reproduction of these 2 million species that we have discovered so far. There's not really a third mode. Animals and plants and fungi and bacteria are reproducing either sexually or asexually, okay? And mostly asexual. So sexual reproduction is a second thought. It's a more developed way of reproducing, and it has definite advantage, but it's more expensive energetically. I can tell you, cutting to the chase, that sexual reproduction is about twice as expensive energetically. In other words, organisms will spend twice the amount of ATPs reproducing sexually than reproducing asexually, all right? because essentially it takes the production of what we call gametes. And you know that gametes are the sex cells. Whereas in asexual reproduction, there is no production of gametes. So that energy that is spent in producing gametes through a process known as meiosis or gametogenesis that we'll cover next week, that energy is saved when an organism reproduces asexually. And again, I'm talking about not just animals, but also plants, fungi, and bacteria, okay? All right, so these are the two modes. And to get it out of the way, I'm gonna give you some examples of asexual reproduction first, of which we don't really participate, except possibly in one exception that I'll tell you about later in the course. That one is just my opinion, but I think that it's one particular mode of asexual reproduction that we undergo in humans, but it's controversial. Anyway, we'll get to it toward the end of this course. So there are many different types or examples of asexual reproduction. In contrast with sexual reproduction, which is only one way, essentially the fusion of gametes, otherwise known as fertilization. But for asexual, for non-sexual reproduction, there are different types or examples, and I've listed most of them here. Binary fission, budding, vegetative propagation, sporogenesis, fragmentation and regeneration, clones, parthenogenesis, and unicellular mitosis. Okay? Now, again, for the scientists in the room and online, you've seen this before at the bachelor level, so bear with me. These are some examples. Binary fission, for example, mostly bacteria. You see individual bacteria here, and then this one is splitting in half. So binary fission is just a fancy word for splitting in half. Fission is division, fusion is union, fusion is union, fission is division. Binary is a reference to two, to splitting in half. Here is one beginning, back here beginning to split in the middle. This one is more advanced. This other one is almost done in the split, and so forth. So that's binary fission. It happens a lot in bacteria. That's their primary mode of reproduction. You notice it's a very fast way of reproducing because essentially it's a duplication. Quantitatively, it's a duplication of the bacteria. And that's why bacterial infections tend to grow quickly. Under ideal conditions, the average bacterium will reproduce, will fission binarily, right? How often? Do you know the time frame? Yes, about 20 minutes actually. Under ideal conditions, about 20 minutes, right? Under ideal conditions, which means temperature, nutrition, and all that is maximized. So every 20 minutes, imagine there's a duplication in the population. That means that it's a geometric progression, right? From one bacterium, we get 2 in 20 minutes. And then in 40 minutes, we have 4. And in 60 minutes, we have 8. So it's a very fast, it's a geometric progression. It's actually this doubling factor, so that in a few hours, we have thousands, and in a few more hours, we actually have millions of bacteria from one single bacterium, okay? And that's why infections have to be addressed fast, soon. As soon as there's a redness, there's a possibility of an infection, and so it has to be addressed quickly. Thanks be to God that in most natural conditions, they're not ideal conditions, all right? In the lab, we can mimic ideal conditions for bacteria, but typically not in the natural world. And that's to our advantage and to the bacteria's disadvantage. Anyway, that's binary fission. Said too much about it. It also happens in other microorganisms like this grab bag of the protozoans. Remember the protozoans, all right? These are paramecia, one paramecium, several paramecia. You can see that they're splitting here in half, right? What needs to happen, of course, is the organelles within have to duplicate before splitting. And so particularly the nucleus needs to duplicate, and then the split will happen. All right, here's an amoeba that has been time frame photography. These are actual photographs, micro photographs, and you can see how the amoeba is splitting into two identical. So these are really all different examples of clones because they come from the same individual, and the DNA is essentially the same unless there is an individual mutation on one of the two nuclei, a chance spontaneous mutation, and then that makes the clone almost identical, but not 100% because of the spontaneous mutation that occurred. Moving forward, then another type is called budding. It happens, for example, in Coelenterates, and I had developed a little list here of vocabulary from previous lectures. Coelenterates is a phylum. It's a group of organisms that are all aquatic because they have a soft body and they're mostly water. Their water content is like 95 to 99% water, okay? And they are, for example, hydras, jellyfish, corals. Corals also have, some corals have a skeleton, and they have a hard skeleton or a soft skeleton. The hard skeleton is calcium carbonate and the classical coral that we see on coral reefs. But then there's also soft coral. They're also known as gorgonians or alcinarians. And their skeleton is protein instead of calcium carbonate. So they sway with the tides and with the motion of the currents. And so, for example, some soft corals are sea whips and sea fans. Let's look at a couple of photos here. It's a good thing about having internet access. Sea fans, for example, these are soft corals. Sea whips are others. Let's see any sea whips. The more stringy. You've seen these on the beach, sometimes wash up, right? And their skeleton is protein instead of calcium carbonate, so they're soft. Anyway, they have these little polyps on them, and the little polyps are actually individual corals, all right? Here's an example behind the sea whip. There's a hard coral, calcium carbonate, and they secrete this calcium carbonate that hardens around them and forms their skeleton. Anyway, those are examples of budding organisms. The Hydra, they're freshwater. They put out this little baby on the side. Eventually, it will clip off here and become a new Hydra, which again is a clone of the adult Hydra. Also happens in fungi, yeast in particular, which is some type of yeast is used for rising bread, right? Brewer's yeast, you've heard of brewer's yeast. Brewer's yeast is used sometimes in the lab because it is truly a eukaryote. In other words, it has a true nucleus, but it's unicellular. It forms colonies, right? Colonies, but these are individual yeast buds, and they put out these little buds that eventually break off, and you have a new yeast there, and then they leave a little scar behind. Is that the scar of a previous bud there? So that's an example of fungi that also reproduce by budding. On the plant side, vegetative propagation is very common in plants. It's a type of asexual reproduction. For example, a tuft of grass, you go out to any lawn and you see these tufts of grass, right? You pull on that tuft and you'll get a string of other tufts that come up, and they're connected by a little connector. And so these are all vegetative propagation. you would trace the origin of that string, wherever it is, and that's the mother tuft. And that mother tuft put out these little shoots sideways, these little horizontal shoots, that when going into the ground, they will produce another tuft of grass further down the line, and so on and so forth. So you get this stringy thing. And that's a type of vegetative propagation. All asexual, no gametes involved, no fertilization. So essentially each one of these tufts of grass, which you cut off that string, and this will grow on its own, all right? These are essentially all clones of the original tuft of grass that generated them. It happens also in some other herbs and small plants. They have a lot of regeneration capacity, and that's vegetative propagation. It actually happens in a few trees also. For example, birch is a typical one, or sorry, I think actually aspen. Sometimes you get a whole colony of aspen. I know this from Colorado, and my brother and sister-in-law lived in Colorado for a time, and in the fall, you have these aspen forests, right, which are just beautiful, the turning of the leaves. But then when you dig under the roots, you actually see that they're all connected by these underground shoots, horizontal, laterally. So really the whole forest is just one huge colony of aspen trees, and they're all clones of each other, all right? The whole forest, they're all clones. And we get that with the Brazilian pepper tree also here, and some other bushes that are tree-like that can do this type of vegetative propagation, okay? Who was, Chris had a question?
Yeah.
Or? Yep.
Yes, sorry, I wanted to go back on the issue of budding.
Yep.
You mentioned that if that organisms that bud, some of their properties are not identical. Actually, it wasn't budding, it was the slide right above it.
Yeah, even binary.
The paramecium.
Right.
So when they clone themselves, their clones are not identical.
They are almost identical, and in fact, they would be identical unless a mutation happens. Remember that mutations happen all the time, all right? There's a background mutation going on, and most mutations are neutral. They don't, because they fall typically into the junk DNA, which is, for example, for humans, 97% of our DNA is what we call junk. In other words, it doesn't code, there's no coding region, there are no exons there. It doesn't code for any protein, all right? And so it's kind of baggage that we have picked up through evolutionary time, including fragments of viruses. Talking about the coronavirus, we have fragments of virus. I think it's about 8 or 10% of our DNA is actually viral fragments that we have integrated over millennia. That may actually be part of the immune system, of how we become healthy, a healthy immune system that is stimulated by the DNA itself that we have within us to produce the antibodies to keep us healthy on a normal situation, all right? Because we're exposed to bacteria and viruses and spores all the time, even just by breathing. But back to this issue, let's say that these two paramecium after the split would be identical, but for the fact that they may be a chance mutation that occurs on either one of the nucleuses, all right? And so if there's a mutation going on, then it's no longer identical. It's 99.999 identical, but not 100%.
And these mutations occur while reproduction is happening?
They can occur anytime.
Okay.
I would say, just kind of thinking out loud, less likely during the actual reproduction process, because remember, we have chromatin and we have chromosomes, right? So when the cell is in interphase, then the DNA is in a chromatin structure, which is a single strand like the spaghetti. I'm actually going to be covering that in a few lectures. And so the DNA is a little more loose, if you will. But then when the cell is going to reproduce, even just straight up mitosis, then that chromatin becomes distinct chromosomes, and it's the maximum condensation. So when there's a maximum condensation or supercoiling of that DNA, there's less likely for a mutation to happen, because essentially nothing can get in there. The agents that cause mutation, UV is one, but also chemicals can cause mutation. But if the DNA is super wound up very tightly, then it's less likely for mutagens to get in there and actually cause the mutation. You follow?
Yes.
But that mutation could have happened just before the cell goes into reproduction, or just after. And then the identical, the 100% identity is compromised. Follow?
Yes.
Okay, but it's kind of it's almost a technical point, because we can say for practical purposes, they're identical. Even if the mutation does happen, but it falls in the non-coding region, who cares? Essentially, they're still identical, because those two nuclei are still making the same proteins.
Okay.
So the coding region was not affected.
So
Right.
would it be right to say that essentially with the cloning, they're identically the same? It's just some sort of mutation could happen after.
Yes.
Okay.
Correct.
Gotcha.
Yeah. So we talk about functionally, even if two nuclei, right, from the same individual after fission or one of these divisions after sexual reproduction, the two nuclei, even if they do have mutations, if they're in the non-coding region, functionally, those two nuclei are identical, because functionally means that they're producing the same proteins. Hmm?
Got it. All right. Thank you.
Mm-hmm. Sure. Oh. Okay, so another example now for fungi is called sporogenesis. And actually many fungi and plants exhibit this alternating cycle of reproduction. It's called an alternating cycle of reproduction, whereby one part of the cycle is asexual and the other part is sexual. And this occurs mostly in fungi and plants. And here is one example whereby spores, we start with the spore of fungi through mitosis, right? Or they will produce the individual gametophytes, whether it's the male or the female, or they can actually be isogametes, that they don't have a male and female, they're just gametes that are haploid, meaning that they don't have chromosome pairs, they just have single number of chromosomes. And when those gametes fuse, then they produce the diploid organism, the other, and then this would be the sexual part of the reproduction, and then that diploid organism will produce spores, and these spores are then diploid. So this is the asexual cycle, asexual part of the cycle of the alternation of generation. It's called alternation of generation, okay? And this is very typical of fungi, and also some plants undergo this alternation of generation. So that's another example. Sporogenesis is another example of asexual reproduction. It also happens in mosses and ferns, like I have here. Back to animals, there's another process called fragmentation and regeneration. This is interesting because a number of animals can actually be cut into pieces, literally, and then each one of those pieces will regrow a new organism, right? Depending on what the piece is, what part of the animal it is. For example, in planarians and flatworms in general, you can actually cut them into several pieces. Here, there's a planarian that has been cut into 3 pieces: the head, the trunk, and the tail region. The head will develop the rest of the body. The trunk will also grow a new head and a new tail. And the tail will also grow a trunk and head back into individual planaria that are functional. Excuse me. So we get 3 from 1 here, depending on how the animal is cut up. And this is fairly typical for flatworms. because they have a lot of regeneration power. Essentially, they have a lot of stem cells in them, okay? It also happens in echinoderms. Echinoderms are starfish, sea urchins, and sea cucumbers, and sea fans, other groups of animals. Basically, they're classified as echinoderms, which means that they have a dermal layer, a layer of skin covering their spines, but it mostly happens in starfish, okay? Whereby if a starfish loses a leg, it will grow back that leg. Typically, the leg doesn't grow back as large as the other legs, a little similar to when a lizard loses its tail. It will grow back like a stump of a tail, but not the full tail, generally depending on the species. But not only that, the leg that was broken off may grow the other legs and become a new starfish in its own right, depending on where the leg actually broke. If the leg that broke off contains part of what is known as the central disc. The central disc, if you draw an imaginary circle touching the base of all the legs, all right, or the angle where the leg bends, you draw an imaginary circle there, that's called the central disc. If a leg cut off contains part of the central disc, then that leg will grow back the other legs and become a new individual. What does that tell you? It tells you that basically the stem cells are in the central disc there, okay? It has a lot of regeneration power. So this is another example, and it's known as fragmentation and regeneration. Question.
When it comes to the new individual from the fragmentation,
yep.
that individual is still fully reproductive.
Yes. As long as it has, again, part of that central disc, that individual is reproductive. And by the way, you know, starfish echinoderms also reproduce sexually. So here's an example where you have an organism with a capacity for asexual reproduction and sexual in the animal kingdom, because starfish and sea urchins and sea cucumbers, all these echinoderms, they also put out gametes. So you also have male and female starfish and so forth and sea urchins, and they put out gametes that float in the water and are in the water column, and they it's external fertilization, but they also reproduce sexually, okay? But this is an example of asexual. And the fact that they can, that a fragmented leg like this that regrew its other legs and puts out gametes, that's an example that it is a functional adult reproduction-wise.
Thank you.
Mm-hmm. Another example is natural clones, as in monozygotic twins. There are two types of twins. If you remember from, I think it was the Ashley girls, remember last semester? That they were identical twins or almost identical twin, 99.99% identical. Just recalling, for example, they both had freckles, but they had a different pattern of freckles in their face. That means they're not actually just a mirror image of each other, right? Okay, so anyway, those are monozygotic twins, right? I suppose, what is the other type of twin? Non-identical twins, and they're dizygotic, D-I, remember? Dizygotic meaning two zygotes. In other words, dizygotic twins come from two zygotes, meaning two fertilized eggs, meaning that that woman or that animal, mammal, whatever, produced two eggs, matured two eggs in that cycle, in that ovulation cycle. And of course, because there are millions of sperm around, those two eggs got fertilized and two individual embryos developed from there. Those are dizygotic. And they're not identical because essentially each one of the eggs had its own DNA material and each one of the sperm had its own DNA material also, okay? And that's why dizygotic twins can be either boy-boy, boy-girl, or girl-girl, right? You have the option. But in monozygotic twins, mono stands for single, one zygote, meaning one fertilized egg, meaning one egg fertilized by one sperm, and then sometime after the fertilization, the split happened. And in fact, in the humans, that split can actually happen within the first 2 weeks. It's amazing that that window is so large. 14 days, up to 14 days after fertilization, that embryo can split into two, and we get two individual human beings. In this case, they will be identical human beings because they come from a single fertilized egg, right? One egg, one sperm. And at some point, either at the zygote level, like here, or later at the morula or blastula stage that we'll see a little later in this course, the split happened. And then it's just a question of whether they share a common placenta or two placentas, and whether they share a common embryonic sac or two embryonic sacs when the split happened, all right? But up to 14 days of after fertilization in humans, we can have identical twinning, which is just amazing. Anyway, moving forward, another type of asexual reproduction, right? It's parthenogenesis. Again, this is a Latin lecture day. Partheno is a reference to virginity. A parthenote is a virgin, okay? And genesis is origin. So these individuals it's a virgin birth, literally. There are some species, it occurs mostly in reptiles and some fish, some lizards and some snakes. Their eggs get stimulated into undergoing mitosis without a sperm. Typically, it could be either temperature, but most likely it's some kind of a chemical, all right, that stimulates the egg into undergoing mitosis and beginning the embryonic process, but without sperm. It's rare, but it occurs, and these are actual examples of it. There's this lizard, I think it's a European lizard, that reproduces by parthenogenesis. This snake, I think it's called the Breheny Snake. Their eyes are vestigial. They don't see very well because they're always buried on the ground, but they're true snakes, they're not worms. They look like earthworms, but they're not. They're actual snakes. And then the dogfish is a type of a shark. Also can reproduce parthenogenically. That means that all the individuals, these adults in the species, are they male or female? They're all female because they're producing eggs. There's no male, okay? It's a rare special adaptation, but it does occur. So they're all females, and I guess it would have to be, they become diploid at the time of the stimulation in the embryonic stage. So here they start as haploid, the gamete, the egg, but once it starts the embryonic development, the chromosomes duplicate, and so the adult will be diploid. Okay, finally, the last one is the most common one by far. It is just good old mitosis. Okay? Mitosis is going on all the time in our cells, in any multicellular organism, and even unicellular organism. We can think of binary fission, and we can think of that splitting of protozoans as a type of mitosis. So mitosis is the universal mode of reproduction for cells in the world, in every species that we have found so far, that we have seen the cells reproducing. They reproduce by mitosis. So we can talk about mitosis, your tosis, everybody's tosis is undergoing mitosis, okay? And here is a plant cell. How do I know that this is plant tissue? Look at the geometry of the cells. Okay, you see these little boxes? They're all rectangulars,
The cell.
boxy. Okay, so these cells, the fact that they're boxy like this means that in addition to having a cell membrane, which is soft, they also have a cell wall, which is hard, kind of like a balloon inside a shoe case, all right, or a shoe box. And so if they have a cell wall, then they're plants. And they're nicely stained, and this is probably a single layer of tissue, most likely onion skin, right? So when you peel an onion, and you get that thin layer in between the other thicker layers of the onion, there's a thin layer there that is a single cell layer of onion, and they're very easy to stain with some kind of a DAPI stain or some kind of a nuclear stain that stains the DNA and not the rest of the cytoplasm. So basically you're seeing the DNA in different stages of reproduction, okay? So remember prophase, metaphase, anaphase, and telophase, the 4 phases of the mitosis of the cell reproducing asexually. First, you have a cell like this one that is not reproducing yet, right? And so you can see the nucleus and the nucleolus, which is this thicker, is a more dense region of DNA. That's called the nucleolus, right? So this cell, we say that this cell is an interphase. It's an interphase because it's not reproducing. Then at some point, the chromatin is going to start condensing into distinct chromosomes, but the nucleus is still there. And then at some point, the nuclear membrane is going to start disintegrating, and the chromosomes will start lining up at the equator. So prophase with the distinct chromosomes, then they line up at the equator, metaphase. And then the third stage is when the distinct chromosomes are pulled. These are chromosome pairs, right? Because the pairing went on before the nuclear membrane was dissolved. Now you have chromosome pairs lining up here at the equator of the cell. Then the next phase is that each one of those pairs, each individual in the pair is going to migrate to the opposite end, to the opposite pole of the cell. And here's the migration, anaphase. And then finally, when they have fully migrated, and then the nuclear membrane will begin to condense again around each one of these 2 clusters of chromosomes. That's telophase, maybe this one here. And then finally, a new wall, a new membrane by pinching in the middle. And then a new wall is built in between the 2 cells, and you get the 2 identical cells in what is known as cytokinesis. So karyokinesis is the splitting of the nucleus, and cytokinesis is the splitting of the cytoplasm itself. Okay. So in this one slide, in this one micrograph, you can see all the different stages of mitosis going on in different cells as the process is going on, okay? All right, let me see, we're at slide number 12 out of 29. We'll go forward a little bit and then we'll take a break. I don't expect you to know in luxury of detail the 4 phases of mitosis and so forth, but just know that there is a duplication. So 2 issues. First, there's a duplication of the DNA in the original nucleus. Excuse me. So duplication of the DNA in the original nucleus, so that when those chromosomes migrate to the opposite end, the 2 daughter cells end up with the same amount of DNA, the same number of chromosomes that the original mother cell, okay? And that duplication has to occur at the beginning of the reproductive cycle. It's also known as DNA synthesis or DNA replication. We'll get to that a little later down. For now, I want to address another issue of, I mentioned that our cells are undergoing mitosis all the time, from every few hours to every few years. But we can say that essentially we don't have any of the original cells that we were born with in our body. Essentially, all of our cells in all of our tissues, in all of our organs have been replaced over a period of years because of this process of mitosis, okay? So why is it that a cell needs to split? It's a surface to volume ratio issue. And I want you to consider this for a moment because this points to one aspect of nature, that nature is very mechanical, Just like when Meyer talked about there being no saltation in the fossil record, no saltation meaning that we don't just go spontaneously from one species to another. There's always some intermediate species. It's just that because of the paucity of the fossil record, because of the scarcity of the fossils, we haven't found, or there may not be a representative intermediate, all right? But there's no saltation, there's no jumping, leaving gaps in between. That points to the mechanicity, to the fact that evolution at the grand scale is mechanical. There's something going on organically, physically, or the same at the microscopic level with this issue of mitosis or cellular division. One way or another, you can take all types of asexual reproduction is a type of mitosis that is going on at the cellular level, all right? And it is this issue of volume to surface ratio. Let's consider it. A cell, any cell, is three-dimensional. There's a cytoplasm, there's a nucleus, so there's a volume. But the surface of the cell, right, is two-dimensional because it's a surface. It's a rounded surface. It's like the skin of the balloon. If we cut up the balloon into segments, we can lay out that skin of the balloon flat on top of a table, right? But the balloon with its integrity is three-dimensional. There's a volume. Okay, so as the cell grows, as the cell is metabolizing, the cell is also growing. It's not only absorbing water, it's also absorbing nutrients, and there are chemical reactions going on. So gradually, as any cell is metabolizing, meaning staying alive, the cell is growing, all right? But you notice that the ratio of growth is different for a surface than for a volume. Because if I double the surface, then that's S squared. Let's say that S represents surface, okay? If I double the surface, because it's two-dimensional, it's S squared. But if I double the volume, represented by V, then it's V cubed, because it's three-dimensional. What does that mean? It means that the cell is cut up in this predicament that as it grows, the volume grows larger than the surface, okay? And so really what happens literally is that the cell membrane becomes too taut. It's like blowing up a balloon, but with water, or inflating a balloon with water. At some point, that skin will stretch and stretch, but it's got a limit, all right? It has a limit. And if we continue to fill that balloon with water or liquid, it's gonna burst, okay? And so this mitosis is a way of redistributing the surface and the volume to cut down essentially on the volume, all right, to cut down on volume. Because consider, when the cell splits, right, the surface is split in half, but the volume is also split in half. But because there's more volume than surface, then more volume was split. You follow? More volume was split. And so essentially by the cell splitting like this in mitosis, the two daughter cells regain the original size of the mother cell. And then each one of them will start growing as they metabolize, if they are alive, and will accumulate surface and volume, but will accumulate proportionally more volume than surface. So they're destined irrevocably, if they want to stay alive, to split again and to reduce by half both the volume and the surface. But in doing that reduction, in doing that splitting, that halving, the one that loses out, if you will, more is the volume, because the volume, there's more volume that gets split than surface that gets split. I don't know how else to explain it, but basically that's the physics of it, all right? And that's the reason for mitosis. It's really for allow cells to continue to stay alive and functional. But in that staying alive, they irrevocably have to duplicate. So you can see that there's kind of a built-in mechanism for organisms to grow. An organism cannot but grow if an organism aspires to staying alive. And it starts at the cellular level. Remember, cells of similar function make tissues, and tissues of similar function make organs, and organs of similar function make systems, right? Organ systems. So whatever organ we're looking at in the organism, we can take it down to the cellular level. And it's because those cells are duplicating, are undergoing mitosis, that the organ as a whole is growing. That growth is most drastic in the embryonic stage of whatever species, okay, including plants, but most drastic in mammals and us as humans. I don't know if I pointed out in the previous course, but I can mention it now. Once I did the ratio, no, I'm going to hold on to that thought for next lecture, actually, because it will fit better when we look at sexual reproduction and the process of fertilization and embryonic growth. So let me hold on to that one. But basically, this is kind of a the physical dynamics of mitosis, why a cell is kind of condemned to have to split if they want to stay alive, all right? All right, this could actually be a good time to break, because now we're gonna go into DNA and some of the genetics of the DNA, the structure and function. So we look at DNA structure and function, following that principle of form follows function. Did I talk about form follows function the last semester? I don't remember. Did I? I mentioned it, right? The structure of the thing is according to the function of the thing. Okay, good. Good split to go out and thaw out a little bit from the from the freezing winter we're having in here, mini ice age. Any questions at this point? No? Okay, so let's take about 10 minutes. It's 10 to 11, so we'll regroup at 11, all right? Let me pause recording here. Okay, welcome back, everyone. Let's get into the second half of this first lecture on human life bioethics, beginning of human life. Again, to put it in context, we want to understand the natural process of fertilization as it establishes a new individual of whatever species, whether it's an animal or a plant, because plants also reproduce sexually. In order to understand that process in some level of detail, the process of fertilization, I want us all to be on board, on the same page, if you will, with regards to some basic DNA genetics. So again, we're looking at structure and function. Structure and function, this is part of the mechanicity of life as we know it here on Earth, organic life, that there is a mechanical basis to organic life, essentially what we call metabolism, which is sophisticated chemical reactions occurring within a cell. So again, structure and function, okay? And this also because we want to speak with competence, right? There are many opinions about many topics today, but one thing is to have an opinion and the other thing is to have an informed opinion. And so we want to be competent when we speak about these issues. Especially, you'll see, as we move forward in this course, to understand and to accept the fact that life begins at fertilization has immense implications, bioethically speaking, all right, immense. All right, so again, for having everyone on board at the same page, if you will, going into the cell, we find that inside the cell there are these organelles, all right, and I think I had another word here on the vocabulary, organelle, right? So you can think of organelles as little tiny organs that have a particular function inside the cell. So again, all based on this form follows function thing. The cell within, being alive, also has its own organs to keep it alive. And these organs, because they're within a cell and they're tiny, they're called organelles. And the most prominent organelle in a cell is going to be the nucleus, generally. And basically the nucleus is a bag of DNA. And the nuclear membrane is also a double membrane, just like the cellular membrane is a double membrane. There are other organelles in the cell that are not the nucleus. For example, the mitochondria that we saw last semester, the chloroplast, which holds chlorophyll, which is used in photosynthesis, a major chemical reaction in nature that is actually the beginning of life, in the sense that it produces the energy, it transfers, photosynthesis transfers light energy into chemical energy, and that chemical energy is the one that is used for metabolism in every other cell, including plant cells, which we know as cellular respiration. There are also ribosomes, there's Golgi apparatus, many other organelles, lysosomes, etc., that are inside the cell. Okay, but I'm focusing on the nucleus now because of the DNA. Within the nucleus, we have the chromosomes, and the chromosomes are made up by the DNA itself, also known as a double helix. Double helix is just a fancy word for a twisted ladder. Twisted ladder, so the ladder has rungs or steps, and it has two rails. So we twist the rail on each side in the opposite direction, and we get a spiral, a double spiral. That also makes DNA very stable, in contrast with the RNA, which is a single rail, and therefore the bases, the nucleotides are at times exposed, and so they overlap and they tend to stabilize with each other, twisting the RNA molecule in things that are called hairpins and everything else. So RNA is much more unstable than DNA. Retrovirus are RNA virus, and the coronavirus is a retrovirus. They're RNA viruses, and that's why they mutate. You may have heard of the UK variant recently, right? More infectious, but seems to be, thank God, less deadly, but more infectious. So this virus has mutated several times already, ever since it left Wuhan a year ago, because it's RNA that is less stable than DNA. DNA is actually a very, very stable molecule. It is amazing. You can call it the molecular wisdom, because at the molecular level, what DNA does is it stores information. And it stores information in a chemical way by simply combining in different patterns four bases which are actually two base pairs. Two base pairs. I'll get to that in a minute. Let's look a little bit more at the overall structure of DNA itself. You heard me mention the word chromatin earlier. And chromatin is the state of DNA inside the nucleus when the cell is in interphase. Interphase meaning that the cell is not reproducing. If the cell is not reproducing, then the cell is doing what? Metabolizing, right? It's being alive, it's metabolizing. So when the cell is metabolizing, not reproducing, the DNA is in chromatin, and the cell itself is what is known as interphase. Interphase in between phases of reproduction. When the cell is going to undergo cell division, then this chromatin, which again, you can think of it as one single spaghetti string inside a bowl. All right, you got a bowl of spaghetti, you got one single spaghetti string, very long. When the cell is going to reproduce, then that chromatin condenses even further. It coils upon itself. It twists more and more upon itself. Think of a rubber band that you're twisting on both ends. And so it becomes more condensed, the maximum condensation possible, and that's called supercoiling, all right? And then at that point, we can actually see the distinct chromosomes. It means also that the chromatin splits, in our case, into how many pieces? In the human, think of the question, chromatin, our cells at interphase. Now, any one of our cells is undergoing reproduction, mitosis at any given moment, right? That chromatin, that string of spaghetti, that single strand of spaghetti is going to split into how many pieces? What's the number of chromosomes in the human is what I'm asking. Number of chromosomes in the human species. 23 pairs, total individual number of chromosomes, 46. So it means that this spaghetti of chromatin is going to split into 46 chromosomes, 46 pieces, fragments. Each one will be a chromosome, distinct. And where it splits, it always splits at the same place. It's not just taking a piece of spaghetti and chopping it indiscriminately, you know, wherever I want or wherever I feel like it. No, it splits in particular places so that chromosome 1 in you is the same, has the same genes and the same coding region and the same non-coding region that chromosome 1 in me and than in anyone else in the world. And 2 and 3 and 4 all the way to chromosome 22, and then the X and the Y chromosomes. So you see that they're discrete regions and they're always the same region in every single chromatin. Imagine the mechanism, the regulatory mechanism for that to happen. Truly, I mean, it's, were it not for the fact that it's an actual mechanical thing, it would be miraculous. You know, that every single cell, just in one individual like us, we have trillions of cells that are undergoing mitosis right now. Now, and then we have so many billions of people throughout the world, you know, so billions times trillions. And every single time that that chromatin splits, it splits in exactly the same place to make 46 discrete chromosomes. I'm talking about normal, right? I'm talking about on average normal because there will be the abnormalities. At any rate, we can go from chromatin to chromosome by condensation and by going from interphase into the reproductive cycle of the cell. Let's drill a little bit into the structure of the DNA itself, simply because I want you basically, what I'm doing here is I'm taking it down to where is the genetic information stored, right? You know that DNA has coding regions and non-coding regions. What do we mean by coding regions? Coding region means essentially a gene. In other words, a region of DNA that codes for a protein. And by the way, we're going to look at 3 functions of DNA, 3 functions that have sophisticated words, and I'll explain them. Replication, transcription, and translation. So these are the 3 main functions of DNA, and that's how the DNA regulates the function of a cell. Okay? So the DNA, you can think of it analogically, is like the brain of the cell. Without ever leaving the nucleus, the DNA itself. And it does that by these 3 processes: replication, transcription, and translation. Okay? So in order to understand those 3 functions, we need to understand a little more detail of the structure of the DNA. So here it is in luxury of detail. Here's the double helix, right? That has been unwound and flattened artificially on a flat surface so that now we have a 2-dimensional model here. And you notice, again, with the big picture first, you got 2 rails represented in yellow. And then you have steps, 1, 2, 3, 4, 5 steps here, or rungs in between the rails, right? Okay. Now, you notice that there are different colors, and there are 4 basic colors. You got blue, red, purple, and green. And each one of these colors represents a different base. So these bases are also known as nucleotides. And there are 4 bases. The 4 bases are adenine, thymine, guanine, and cytosine. An easy way to remember that is when we put them in capital letters, when we capitalize them, capital A, capital T, capital G, and capital C. They're always in pairs. So adenine always pairs with thymine, and guanine always pairs with cytosine, all right? And again, there's a structural reason for that. And I'll tell you in a minute. But basically, an easy way to remember this is that in the capital letters, the straight letters pair and the curved letters pair. So A and T are straight, and G and C are curved. So the straight pairs with straight, and curve pairs with curve. Just one mnemonic for remembering that A, T, C, G is the base pairing, all right? Why is it that they pair this way? Well, it turns out because some of these are 2-ring molecules and some are 3-ring molecules. They take up more space. And so in order to maintain the 2 rails parallel, the pairing has to be this way. A 2-ring with a 3-ring and a 2-ring with a 3-ring. If you put 2 rings with 2 rings, then the DNA is going to kink in. The rails are not going to be parallel. They're going to kink in. And if you put 3 rings with 3 rings, then the rail is going to bulge out, you see, and the rails will not be parallel. So the reason for this particular pairing is because you have a 3-ring molecule paired with a 2-ring molecule, and the same with C and G. You have 3 rings paired with 2 rings, and that keeps the rails parallel. It's part of the stability of the DNA at the molecular level. All right? All right. Next thing you notice on the model is that the letters on the left-hand side are right side up, but the letters on the right-hand side are upside down, okay? That is called, so the rails are parallel, but they're also anti-parallel. In other words, they're going in the opposite direction, okay? So it's not just parallel, it's called anti-parallel, meaning that one strand, let's say the left strand is read from the bottom up, and then the opposite strand is read from the top down. But they are complementary, and that's the important thing. They're complementary. In other words, if I give you just one set of bases, one sequence of bases, you can tell me what the opposite base is, and we can complement the opposite strand. The 2 strands are called, one is called the leading strand, the other one is called the lagging strand. No need to worry about those terms, but basically, if you give me the leading strand sequence, I can tell you what the complement is by this base pairing. And the same, if I give you the lagging strand sequence, you can complement it with the leading strand because of the base pairing, okay? All that was deciphered, and the big geniuses in this were the 2 big names of Watson and Crick in the 50s, where they deciphered finally the structure of DNA. There were several labs throughout the world going after this. Some were here in the States, in California, New York, and then the others in UK and India, other places in the world, but the ones who beat the race were Watson and Crick. Watson was the graduate student of Francis Crick. He was the PI in the lab at King's College in the UK. And Watson was a graduate student from over here from Indiana who had gone over there to do his internship, okay? At any rate, this is the elucidation of the chemical structure of DNA. in the 50s, and then in the 60s, the actual coding regions were deciphered, and the triplet code was finally deciphered. So we get the actual genetic code, which I'll show you at the bottom, that little table that you see with all the letters. So to do the transcription and translation into protein. So we can go from DNA to protein by way of RNA, the messenger RNA. Okay, here we're still with the structure of DNA. Now we need to zoom out. Oh, I'm sorry. The information is actually held in the base pairing. This is where the information is stored at the molecular level, all right? The DNA information, the genetics, the genes, they're stored in the particular sequence of base pairs. That's where the information is stored, all right? So you can think of this analogically with a computer. These are the bytes. These are the bytes in a computer program where the information is actually stored. Let's zoom out now and look at the coiling issue. We start, because it's a coiling on top of a coiling on top of a coiling, right? We start with the DNA strand, which is the double helix. So that's the first level of coiling, if you will, of twisting, is the twisted ladder, literally. Then that twisted ladder, that long string, you can think of it as a rope, winds around some proteins that are little balls of proteins. Those proteins are called histones. And the standard ball of proteins is 8 histones, actually 4 pairs of histones, okay? 2A, 2B, 3, and 4 are their names. Don't have to worry about it. But basically, these are little balls of proteins, and there are 8 balls, and there are 4 pairs of them, and they're called histones. And these histone proteins are some of the most conserved proteins in the world, meaning that they hold on to their sequence. Basically, it's the same sequence throughout the animal and the plant kingdom and so forth. What does that mean? That means that these histone proteins must have been developed early on in the process of evolution, so that most organisms, their DNA winds around histone proteins. Okay, and like I mentioned, there are 4 pairs of them, 2A, 2B, 3, and 4 are they're referred by those numbers, and they're highly conserved. For example, histone protein 2A of us is essentially identical to histone protein 2A in the fly, all right, and so forth. So this is before they develop, before divergence. So where is 1? It kind of begs the question, where is histone protein 1? Histone protein 1 is the linker, is in between. And it stabilizes that little string that goes from one nucleosome to the next. So backtracking a little bit, you have this cluster of histone proteins, right? The 4 pairs, the 8 balls together. And the DNA will wind around it almost 2 times. The DNA helix, double helix, will wind around almost 2 times, 1.7 times to be exact. And it's kind of like a yo-yo, like the string that winds around a yo-yo twice, okay? And then it moves on to another cluster of histones and winds around that, and then moves on to another one and winds up around that, so that in the end, you get this effect of what is known as beads on a string, beads on a string. Kind of think of a necklace, a pearl necklace, all right? You get these little beads on a string, and each one of those beads, all right, is known as a nucleosome, and this nucleosome is the functional unit of DNA. Listen to what I'm saying. The nucleosome is a functional unit of DNA. Just like the cell is a functional unit of life, right? Organically speaking, here on Earth, the cell is a functional unit of life, right? The nucleosome is a functional unit of DNA. It is a stable, it's very, very stable complex, and it is a complex of proteins and DNA. The histone proteins, the 8 little histone balls, and almost 2 winds of DNA going around it. So that's the second level of coiling. The first level of coiling is the actual twisted ladder, Second level of coiling is the nucleosome. Then you get these beads on a string, but it doesn't stop there. Those beads wind upon themselves again. So it's like taking a pearl collar, pearl necklace, and winding it, just twisting it at the opposite ends and bringing those ends together, you get another coil. That's a supercoil, all right? So now we got this coiled nucleosome string that is already at the supercoil level, right? Because there's coils upon coils upon coils. And then that coil winds upon itself again, two or three more times. So you get a supercoil on top of a supercoil, and you get this winding effect. In the end, when that is going to be for each chromosome, you have all this supercoiling going on, and that is what is known as DNA compaction. The DNA is being compacted, all right? It's being compacted. So that's DNA compaction. Now, here's a question. If we take out a single chromatin molecule, let's say that the cell is an interphase, and we dip into the nucleus hypothetically with our mind's eye, and with a little tweezer, we pull out the chromatin from one tip, and we stretch it out, all right? Keeping the integrity of the double helix, but unwinding the supercoiling, if you will, and just at the level of the double helix. So the double helix has integrity, but there are no nucleosomes, all right? Just stretched out a lot. How long would that molecule be? And it will be a single molecule, right? And how many atoms? Millions of atoms. But it's a single molecule because it's chromatin, all right? How long, approximately? Well, it's not that long, all right? But it's significantly long because we're talking about a single molecule. It's about 6 feet, 2 meters. So 1 meter, 3 feet is from the tip of my fingers to the opposite shoulder. That's 3 feet or 1 meter, more or less, approximately. So 2 of those, all right? Pretty long, really, for a single molecule, right? That long. How is it possible that 6 feet of a molecule fit inside a nucleus of a cell? Already the cell is microscopic, and the nucleus is inside a microscopic cell, okay? How can we fit 6 feet of a string inside that nucleus? By the compaction, by supercoiling, all right, compaction. And what is the compaction rate? What is the compaction rate? All right, so if I fold this in half, okay, I have compacted this page into half. I've compacted it into 2. So I have 2 surfaces, right? From 1, 2 surfaces. If I compact it again, if I fold it again, I have compacted into how many total pages? 4, right? And that's the second fold. 3-fold, 4 times 2 is 8. So there are 8 little pages in here, right? 8 surfaces, and I have folded, how many times? Okay, 3 times. Okay, so compact again, 4 times, 16 pages, 5 times folding, 32. 6 time folding, 6-fold. There's 64 little surfaces here, right? Okay, I challenge any one of you to fold that again without using any tools. So we have done what, 6-fold, right? 1, 2, 3, 4, 5, and 6. Yeah, there's no magic, right? There's no rabbit out of the hat. So that's a 6-fold compaction. To get that 6 feet of DNA into the nucleus, there's a 10,000-fold compaction going on. 10,000-fold, all right? So all this to say that this compaction is highly efficient, super highly efficient to get a 6-ring molecule inside a super microscopic compartment, which is the nucleus of the cell. Highly efficient. What does that do to the DNA itself, to the sequence? It essentially shuts it down. It shuts it down. So the replication machinery or the transcription machinery has a hard time getting in there because it's all wound up tightly, all right? And is that a good thing or a bad thing? Well, think about this. Each one of our chromatin, of our DNA in each one of our cells has the full 23 pairs of chromosomes, the full 46 chromosomes, right? So all of our genes, it's estimated we have about 10 to 20,000 genes. So all of those genes are inside each one of our cells, in every part of our body, in every organ and tissue of our body. And yet our organs and tissues have different functions, right? The brain doesn't do what the liver does, and the liver doesn't do what the kidneys does, and the kidneys don't do what the heart does, et cetera, et cetera. So each organ has a specific function, which is regulated ultimately by the metabolism of that tissue, of those cells that are there, right? So liver cells do liver things, and brain cells do brain things, and so forth, meaning that organically, the genes that are being deciphered, the genes that are being expressed are either brain genes or liver genes or kidney genes or heart genes, et cetera, et cetera. And so it means, but we have every single gene in every single cell of our body. So imagine if the DNA were more unwound and more open up so that the transcription machinery, which is the one that's going to lead to the protein synthesis, were able to read all of the genes in every single cell. We would get an overwhelming expression of every gene in every cell of our body. And it would be essentially chaos, because we would have every cell of our body expressing every gene that we have for every metabolism, for every pathway, metabolic pathway that the proteins are regulating, et cetera, et cetera. And it would be totally chaotic. We would not be able to have life, all right? And so life really, at the organic level, is an orderly process. And it is ordered because the DNA is mostly shut down. So most of our DNA is shut down most of the time in most of our cells. And that is a good thing. That's how we can actually have life and metabolism in an orderly fashion, all right? So what happens functionally? The liver cells, only the liver genes, only the region that codes for the liver genes are the ones that are going to be opened up enough for the transcription machinery to get in there and read, decipher, express those liver genes, right? And so on and so forth. And you can make the same argument for all of the tissues of our body so that we have a functional whole organism instead of just chaos. All because of the structure and the function of DNA. And this is all regulated also in itself is regulated by proteins, okay? So, and these are the regulatory proteins. Okay. I talked about the anti-parallel issue, the fact that they go back and forth. Hydrogen bonds, don't worry about it. Some are double. The A and the T bonds are double bonds, and the C and G bonds between the base pairs, the bearing, is a triple bond. Too much detail, not to worry about it. Those are hydrogen bonds. When the DNA splits, those bonds are broken so that the DNA's molecule actually unzips at the middle. And the base pairing become unpaired. Okay, one last diagram here on the detail of the replication machinery itself. All right. Remember I mentioned in passing that when the cell is going to reproduce, before it reproduces, the DNA in that cell, in that nucleus, has to duplicate, Why is that? Why is it that the DNA has to duplicate? Go back here for a moment. Here's a cell in interphase, right? prophase, anaphase, telophase, and then the two daughter cells. You notice that, let's say that these are two daughter cells here, just from the size, that came from an original mother cell that looked like this, okay? The genes that are in each one of these daughter cells. First of all, these two daughter cells, do they have the same chromosome? Are they essentially identical? They're essentially identical, right? Except for possible mutations that may have occurred at some point. All right, so they're essentially identical. The DNA here and the DNA here. And how about the DNA here and the DNA here of the mother cell? Do the daughter cells have the same DNA as the mother cells? I'm going to say yes. Daughter cells have the same, each daughter cell has the same DNA as the mother cell? Yes? No? Unsure? The rest don't care? They do, they have to, they're clones. They have to. Now, how did that happen? Again, there's no magic, there's no magic wand, there's no pulling rabbits out of a hat, okay? There has to be a mechanical explanation for the fact that the daughter cells have the same DNA that the mother cell, but I have two daughter cells and one mother cell. What had to happen to that DNA before it went to anaphase? It had to duplicate. The DNA had to duplicate. Okay, so temporarily, when we, what is diploid? Have I talked about diploid before? That we have chromosome pairs, right? So it's called diploid, a cell is diploid. And we also call it 2N. I don't know if I'm talking about 2N here. I think in the next lecture, I talk about 2N. Anyway, the DNA has to duplicate before the cell splits, okay? And that DNA duplication, we call it scientifically replication. Again, you know, in science, we don't want people to understand what we're talking about, so we change the words. But DNA replication is a duplication of the DNA. And it happens in a stepwise process, again, because it's very mechanical. This is all regulated by proteins, by enzymes. And that's what I was talking about, the splitting of the DNA molecule. The double helix is split in half, literally. What happens is those hydrogen bonds are cut, right? Hydrogen bonds are cut so that the base pairs are unpaired. Unzip is literally, it's like unzipping this, this DNA molecule down the middle. So what happens then, once the molecule is unzipped, now you have the bases, the individual bases are just sticking out, begging for a pairing, begging for a pair, all right? To be complemented, on both strands, on the leading strand and on the lagging strand. So these nucleotides, like the word says, they're free nucleotides. They're not free that they don't cost any money. They cost a lot of money. They cost basically food, because when we eat, part of what we eat is nucleotides, okay? And so some nucleotides are taken and some nucleotides are synthesized by the body. But the point is that the cell has a surplus, of nucleotides that are floating around. And those nucleotides are then used to complement the two strands that remain unzipped, all right? So those bases that remain hanging out, waiting for a complement, are complemented by what is known as the replication machinery. These are proteins, they're known as polymerases, all right? And they're protein complexes, three-dimensional processes, complexes that are going through the DNA at lightning speed. They do this very, very fast, in fractions of a second, because there's exposure. And with exposure is tremendous danger, okay? These nucleotides, when they're exposed like this, they're very subject to mutation. And so the replication machinery, the replication process is very fast, in fractions of a second. What happens when we do things very fast? We make mistakes. all right? On the average, I think it's 1 every 10,000 or so, there's a mismatch. The wrong nucleotide is put in, the wrong base is put in, so there's a mismatch on base pair. That is considered a mutation. So then there's another machine that comes behind this one to fix the errors that were done on the first run. Okay, but the first run is done very fast to get the DNA back to its base pairing, to stabilize the DNA molecule so that it is no longer exposed to mutations and to mismatches and to errors, all right? Sequential error. Why? Because this is the core, this is the information is stored here. And if there's a mistake in the information, that may code for the wrong protein. It may actually destroy the protein. If this is in the coding region and this is the active site, this codes for the active site of the protein, we have functionally destroyed that protein because the active site now is not going to be functional. All right, and it could come down to 1 or 2 or 3 base pairs. These are point mutations, and sometimes in genetics, these cause diseases that can be traced down to a single point mutation of 1 mismatch of a base pair, all right? Seldom, but it does happen, and these diseases are out there. So the replication machinery is very fast to minimize exposure, but in being fast, the downside is that it does make some mistakes. So there's a second machinery that comes through afterwards, correcting most of those mistakes, like 99.99% of the mistakes. But you know, we're talking about 3 billion base pairs. So some mistakes may actually stay in even after the repair mechanism went through. Sophisticated to the hilt, like you wouldn't believe, all regulated by proteins, regulatory proteins in our body, in every single cell, all right? Okay, so what else? Essentially, replication is a duplication of the DNA, and it happens inside the nucleus just before the cell is going to split, just before the telophase, the prophase. It's at the end of interphase when the cell is undergoing, undergo reproduction. And this is also known as DNA synthesis. DNA synthesis is a third word. So you got duplication, which is the same as replication, as DNA synthesis. It's the same thing. Okay, so that's one process, replication. Now the other process is transcription and translation. When the cell is not reproducing, then the cell is metabolizing, all right? Metabolizing meaning that there are biochemical processes that are going on in the cell all the time. It's a dynamic issue. Metabolism, you know, is subdivided into 2 categories, anabolism and catabolism. Anabolism is building up certain chemicals, and catabolism is breaking down certain chemicals. And so the cell, to metabolize, to be alive, functionally is doing that. It's building up certain molecules and is breaking down other molecules back and forth in a dynamic state. And that's what we call metabolism, and that's what we call life, right? So all that is done by proteins, regulatory proteins and functional proteins. And the proteins are synthesized, are made, produced in the cytoplasm. But the information for that protein, the sequence for that protein, is in the nucleus, in the RNA base pair sequence, right? So somehow that information needs to get out to the cytoplasm for these little machineries, these little ribosomes, which themselves are a combination of protein and RNA, okay? Ribosomal RNA, rRNA. These complexes are the ones that actually synthesize, make the proteins. But they need the information. They need the blueprint for what protein sequence am I going to make. And that protein sequence information comes through an intermediary, which is called messenger RNA. Very unimaginatively, it's a messenger from the DNA to the ribosome. and it's RNA, meaning it's a single strand instead of a double strand. Okay, so mRNA is the one that carries the message, the information in its sequence physically, has the sequence, which is the complementary sequence of the DNA strand, all right? So it's like a complementary, except for one twist, which is instead of having thymine, RNA has uracil, which is a very similar molecule, but one of the radicals is different. So it's called uracil instead of thymine, and therefore the letter that represents it, the acronym is going to be U. So for RNA, it has U instead of T, so it's going to be AUCG, all right? That's RNA, messenger RNA. So the messenger RNA carries the message structurally from the DNA to the ribosome, and then the ribosome functionally reads that sequence, that base sequence, and matches up the building blocks of protein. What are the building blocks of protein? They're called amino acids. And amino acids are particular molecules that are found in nature. There are about 20 amino acids in nature, naturally. Okay. So the amino acids are the complement, the analogous of the nucleotides for DNA. Amino acids for proteins are the analogous of the nucleotides for the DNA or the RNA. Follow? Okay. So these are the basic building blocks of proteins, these amino acids. There are 20 in nature. Let me move forward a moment here. I talked about the genetic code just to show you for a minute. These are the 20 amino acids put here in groups. They're represented in these contraction or abbreviations. For example, PHE is phenylalanine, LEU is leucine, ILE is isoleucine, MET is methionine, valine, tyrosine, proline, histine, glycine, et cetera, they're all NNIene, okay? Aspergine, glutamine, glycine, et cetera. So if you count these little abbreviations here, you're gonna find 20 plus these stop. These are known as stop codons, which is UAA, UAG, UGA, all right? These are stop codons. In other words, this is not an amino acid, a stop amino acid. This is no amino acid. What are the three letters? What are the triplets? The triplets are the nucleotides. It's a nucleotide code, right? So UUU, remember this is RNA. So in the DNA, this would be what? UUU, TTT, right? TTT codes for phenylalanine, and CAU codes for histine, and CGA codes for arginine, etc. And this is what is known as the genetic code. And this is what was deciphered in the 1960s. And this allows us today to read, literally translate any DNA sequence into its equivalent protein sequence. And actually to see if there is a protein sequence or not, right? Because it could be, and then if there is a protein sequence, then that's coding region, which we call exon. But if there's no coding sequence, then it's what we call the junk DNA, the non-coding, which is 97% of our DNA is non-coding, right? And the way to do this table, to build this table, is you need 3 sides out of the 4. You need 3 because a triplet is made up of 3 bases or 3 nucleotides. So here's the first nucleotide, here's the second nucleotide, and here's the third nucleotide. You notice that the first and second nucleotide are single, UCAG, because it's the RNA, it's the mRNA. If we were to go back to the DNA itself, we just substitute this U for a T, no big deal. But you notice that the third triplet, the third letter in the triplet is any one of the 4. Any one of the 4. Anybody know what that's called? That's called wobble. Wobble, it wobbles. In other words, the third letter can vary and it will still code for the same amino acid. For example, again, on the first one here, UUU codes for phenylalanine, but also UUC codes for phenylalanine. So there's a wobble between U and C for phenylalanine, okay? For leucine, UUA or UUG, either one will code for leucine. So there's a AG wobble here. Look at serine. Serine, any one of the 4 bases on the third position, doesn't care, doesn't matter. So the action really is in the first U, or the core, the very core of the information is the first U, first 2 letters of the triplet. The third triplet can wobble back and forth, some more than others, okay? Methylaniline doesn't accept any wobble, has to be UAG. And this is unique. You notice this is unique because typically methylaniline is the first, methionine is the first triplet on a coding region. It's the first one, it's the lead, it's like the locomotive on the train, all right? So when the ribosome sees a methionine, sees an AUG, starts a new strand of protein, a new strand, okay? And that's why it is unique and doesn't tolerate a wobble. But the others do. And this, again, has been established over millions of years in the evolutionary process of the translation from DNA to proteins, okay? But it is universal. So the triplet code, the genetic code, right, which is this one, is universal. Regardless of the organism, if there's going to be protein synthesis, it has to follow this genetic code, all right? So you can imagine the key that this opened. So the big boost, if you will, in molecular genetics last century was precisely in the 50s and the 60s, first in the 50s, in the elucidation of the DNA structure itself, and then about a decade later, the actual function of the coding region. And it comes down to these 3 triplets, these 3 bases in a sequence. We're talking about literally a sequence of 3 bases, right? For example, 2 green and 1 red, or 1 red, 1 yellow and 1 blue, etc. Any one of these 3 triplets and any consecutive bases could make a triplet, all right? We don't know. Until we decipher it, we don't know where the coding region starts. And that's how the information is held structurally in the DNA, which then functionally is interpreted by the ribosome via the messenger RNA. Okay, now there's an intermediate molecule just structurally. Remember that the basic unit of proteins is always the amino acids, right? Look here for a moment. And like I say, there are 20 of these that occur naturally, meaning in nature. Then other amino acids have also been synthesized artificially. Yes, you can buy them in bottles. Maybe it's too big. Yeah, it's a table that has the 20 amino acids, for example, like that. Okay, and you see these are the stick molecule conformation structures. They're classified as polar, non-polar, etc. If they have a charge or no charge. Some are rings, but most are just sequence. And they all have a nitrogen molecule atom embedded into them. And then there's a chain, like a carbohydrate chain that's sticking out. Okay, so these are the 20 amino acids. They're medium-sized molecules on the small side. But the point about this is that these amino acids in a sequence a few dozen or a few hundred, make up this intermediate structure that we call a polypeptide. And that's actually, we say one gene, one polypeptide. In other words, a gene structurally in our DNA typically codes for a polypeptide. And then several polypeptides coming together form the actual protein, the protein complex, right? But the intermediate structure is this polypeptide. Okay, for the non-scientists, hopefully I'm not giving too much detail here that I lose you, but this is the separate mechanism that happens when the cell is not replicating, when the cell is not, I'm sorry, when the DNA is not replicating, is not duplicating, right? Then the cell is metabolizing, meaning that proteins have to be synthesized, meaning that the message, the sequence has to get to the ribosome, and therefore that intermediate, and this is a two-step thing. DNA, when DNA makes RNA, it's called transcription. And then when RNA makes protein, that's called translation. So transcription is with nucleic acid. In other words, from DNA to RNA, that's transcription. And then from RNA to protein, that's translation, okay? So you can think of it this way. For example, if we have a book, a manuscript will be handwritten. Let's say we're writing a book or an article, we write it by hand first. That's the original, let's say that's the DNA. Then we transcribe that into a Word document. Okay, so we're going from the handwritten to the typewritten document. That's a transcription, but it's still the same message that is there. Then we can take that and we wrote that article in English, and then we can translate that article into German. We have translated, we have gone into another, which is the protein. We have left the nucleic acids and now we're dealing with protein, all right? So that two-step thing, transcription from DNA to RNA, and then translation from RNA to protein. Transcription happens inside the nucleus, translation happens inside the cytoplasm, different places. Essentially, when we put it all together, this is known as the central dogma in molecular biology. Like I say, it's a universal process. This happens in every single organism, from bacteria to the largest whale that you can think of that we have been able to study, is this one single process of transcription and translation going from DNA to a functional protein, okay? And that's why it's called the central dogma of biology or molecular biology. Central dogma of molecular biology is this transcription translation. The central dogma of organismal biology and ecology is going to be evolution. So here it is in parts. This is a transcription part from DNA to RNA. And then the translation part from RNA to the polypeptides. Several polypeptides will then make up the actual functional protein. More visualization here, just simply to show, just like the DNA sequence winds upon itself, all right, to form a three-dimensional structure. Proteins also do that. Polypeptides come together. These are individual amino acids here, represented by different colors, and they have two options. The polypeptides either form a coil or a sheet. The coil is called a helix, right? It's a single helix, it's not a double helix like DNA, it's a single helix. And so if the polypeptide coils, twists upon itself in a single helix, it's called an alpha helix. But if the polypeptide folds in a sheet, right, back and forth, folding upon itself, then it forms what is known as a beta sheet, a beta sheet. All right. And then combinations of these helices and sheets, alpha helices and beta sheets, combinations of these form the actual three-dimensional functional protein. Okay. One last step. These three-dimensional proteins then come together to form what is known as a protein complex. And that finally, the protein complex, is the one that is truly functional in metabolism, okay? And that is known as the quaternary structure of the protein. So the primary structure is the actual amino acid sequence, just like it would be for DNA, the nucleotide sequence. Then the folding into either coils or sheets, that's the secondary structure. Then combinations of these helices and sheets form the tertiary structure. But then these proteins coming together into a complex finally form the quaternary structure, which is the actual functional protein. Just one example for this represents hemoglobin. And hemoglobin is made up of 4 identical proteins. Each one of them is called a myoglobin. Okay? And these 4 need to come together in a complex fashion, three-dimensional complex fashion, to form the actual functioning hemoglobin in our red blood cells. What does hemoglobin do functionally in the body? It carries oxygen from the lungs to the tissue, and it carries CO2 from the tissues to the lungs, right? So both. And it's hemoglobin is a quaternary structure of 4 myoglobins, right? And what is holding the 4 myoglobins together at the core? What is known as a heme group. Heme is another fancy word for iron, ferrum. So there's an iron atom core at the center, holding the 4 myoglobins together. And that iron, because it's a metal, the iron can oxidize, right? Another word for oxidizing is rusting. And so when that iron picks up oxygen at the lungs, it oxidizes. And what is the color of rust? It's a reddish color. And that's what's giving the reddish color of the blood. So you notice, again, we have layers of layers of layers, and all this is controlled by regulatory proteins, and this is all part of the metabolism functionality of the cell. Okay, where did I leave off over here earlier? All right, so in the end, we have 2 separate processes. If the cell is going to reproduce, then the DNA first has to duplicate, which is called replication. And that's how we get from 1 DNA strand, we get 2 DNA strands. And the 2 daughter cells end up with the same quantity and quality of DNA, of genes, that was in the original mother cell, okay? All through replication. So that single word is very informative. Replication, DNA replication. If instead the cell is not reproducing, then it's metabolizing, it's staying alive, and it's doing that in a 2-step process of sending the DNA information out to the cytoplasm so that proteins may be synthesized. The functional proteins, the regulatory proteins that are going to be involved in all the pathways of the living of that cell, of that tissue, of that organ. And that 2-step process is transcription, translation, transcription from DNA to RNA, and translation from RNA to protein. That is what we call the central dogma, okay? You may also run across this other phrase, this concept of semi-conservative For replication, replication is semi-conservative. Look at this. Why is it semi-conservative? Has nothing to do with a political party or anything like that, or a platform association. It means that half of the DNA molecule is always conserved or preserved or maintained in the duplicate. You see? Because the complement DNA is being synthesized upon the original template. So the original template is always part of the original DNA molecule. All right? Half. So for one DNA molecule, we get 2, and each one of those 2 has half of the original rail. And then the other rail is the new rail. So that's why it's called semi-conservative, because it conserves or preserves one of the 2 rails in every new synthesis, in every new replication. So replication is semi-conservative. Now you know all these words. Talked about the DNA code already. Redundancy is another one that you run across. Redundancy has to do with the fact that several triplets code for the same amino acid. Several different triplets, precisely because of the wobble. So wobble allows for redundancy. Redundancy means how many, think of this. How many possible combinations do we have of triplets? When we put any three words, 3 letters together, how many possible combinations we can have? We have 64, because it's 3, no, there are 4 letters, so it's 4 cubed, no? 3 layers. So 4 square is 16, and 16 square again is 64. So it's 4 cubed, right? 3 possibilities of combinations of the triplets. So it gives us a total of 64 possible combinations. You notice, if you look at this table, on the triplet code, there's no code repeated. All right, there's no code repeated because there's 64. We have groups of 4, and 4 times 4 is 16, and times 4 again is 64. Now, we only have 20 amino acids. So 64 triplets code for 20 amino acids. That means that more than one triplet codes for any one amino acid. Again, because of the wobble. All right, and the possibilities are anywhere from 1 triplet to all 4 triplets on the 3rd base, coding for the same amino acids. And that is what is called redundancy. In other words, more than 1 triplet DNA may code for the same amino acid. One last thing. We hear about DNA and RNA all the time, but then few people, I think most people have already heard about DNA and RNA, even now more so in the past year because of the RNA virus, okay? But few people actually are able to tell you what those 2 acronyms stand for. So DNA, just for the record, stands for deoxyribonucleic acid, which is a mouthful. And then RNA stands for ribonucleic acid. So there's no deoxy. And the deoxy part is what allows for the double helix as opposed to the single helix. So that's what the acronyms stand for, deoxyribonucleic acid and or ribonucleic acid for RNA. Here are their bases, the complementary base pairs. So you notice that 3 are identical for DNA and RNA, except for thymine, which is uracil in RNA. And I told you it's a single ring, first of all. And you notice that it's essentially the same molecule, except that this radical here is a CH2 group, whereas here it's only a hydrogen. And here also, there's an extra hydrogen to stabilize this nitrogen down here. And that's it. So they're very, very similar, but the active site then is different. So it has a different name. It's actually a different molecule. They're similar, but they're not identical molecules. What else? All right. We're coming to the end here. Kind of a recap for mitosis. Only one cellular split. The cell splits only once. This is important when we look at sexual reproduction next Saturday, because for sexual reproduction, for making gametes, for making sperm and egg, there's a double cellular split, not single. And that process is not called mitosis, it's called meiosis. And it's like a double mitosis, okay, with a twist. So mitosis is a single, simple cellular split, whereby from one cell, we end up with two cells, and the chromosome, the diploid number of chromosomes is maintained. Diploid meaning that there are chromosome pairs. So the mother cell is diploid, has chromosome pairs, and the two daughter cells also have chromosome pairs. We represent that numerically with this little 2n, where n is the number, the single number of chromosomes for that species, okay? So in the human, n is 23, and 2n is 46, right? So when we say just n, what is implied is the coefficient. The coefficient, when I just say n, the coefficient 1 is implied. We don't write it. We don't write 1n. Just like in algebra, if I put a plus b, it's 1a and 1b. But if I put 2a and 2b, then it's 2, right? Okay, so 2n represents diploid chromosome pairs, and 1n, a single n, represents one set of chromosomes unpaired, and that is known as haploid instead of diploid. And all the cells in our body are diploid except one type of cell, which is the one type of cell that is haploid in our bodies. The sex cells, whether we're male or female, right? And those are the sex cells. So the male is going to be sperm, and for the female is going to be the eggs. And it's important, it's essential that they be haploid so that when they fuse, then they restore diploidy. They restore the diploid number, all right? So it's a little anticipation of what we're going to cover in greater detail next lecture, which will be sexual reproduction. So I just leave it at the end with a diagram of mitosis. One more time, one mother cell, chromosome pairs, two daughter cells, eventually end up with chromosome pairs. And there was a temporary duplication here, temporary duplication. So in this cell, temporarily, this cell is 4n, temporarily. This stage here is temporarily 4n because the chromosome pairs are about to split. Okay, that's it. So the DNA genetics, we understand now this little molecule that we just talked about, DNA, there's a lot of information compacted in there, right? Structurally and functionally. But basically, it is the molecule of inheritance. That's where the genetic information is stored. And now we have seen, at least at the molecular level, how that information can either be replicated to produce another cell like itself, or transcribed and translated into a functional protein that will then be in charge of metabolism within that particular cell. Okay. I don't know about you, I'm exhausted. We've got a lot of material in a short time, but I think for most of you, this is just a review of your basic biology or college biology. Anything else? Questions, comments? No? Done?
Just one. Are we going to be using a book for this course?
No, not really. There are a number of references. I mean, really this, this material is pretty basic and it's out there on the internet. I could provide some references. The standard reference is Campbell, but there's so much. Campbell Biology is a big textbook that we use in our bachelor's program, but there's much more information there than is really needed for this course. So
Okay.
there's no textbook as such. I'm providing the material and I'm getting from here and from there, you know, just selective things. But no, bottom line, no particular textbook for the course.
Okay, perfect. Thank you.
You're welcome. Okay, folks, so I'm going to stop the recording now. Thank you once again for being present, and we'll pick it up next Saturday, God willing. Thanks.